ಇಲ್ಲಿ ಸಿಗ್ನಲ್ ಸ್ವಲ್ಪ ಇರಲ್ಲ. ಇರ್ಲಿ ಬಿಡು, ಇರ್ಲಿ.
So, Mr. Mohana, so you can share your presentation.
Okay, sir.
So, you will have 5 minutes of presentation. Okay.
Okay, sir.
Yeah. Please be stick to your timeline. And Dr. T.S. Harsa, you will be next. Okay. Please be ready.
Yes, sir. Yes, sir.
Yeah. Please be ready with your presentation.
Hello, sir.
Yeah. Please share your screen.
Sir, allow for me, sir, for sharing.
Just a minute. Just a minute.
Oh, okay.
Yes, yes. So now you can share your screen.
Okay. Sir, it is visible?
Yeah. Please put it on full screen mode.
Okay, sir.
Yeah. So now you can start.
Okay. Today I am presenting my research work, exploitation of Dictyosterium ehrenbergianum for biodiesel production using sewage-rich pond water isolation and characterization and biodiesel property analysis. As you know, in the past decade, the overconsumption of fossil fuel leads to increase of global warming, acid precipitation, air pollution, ozone depletion due to the emission of many gaseous toxic substrates such as carbon dioxide, nitrogen gas, sulfur dioxide, carbon monoxide, etc. And further, the rapid decrease of fossil fuel followed by rapid decrease of these gaseous elements, it is necessitated to search some alternative fuel from natural sources. Biofuel is one of the renewable, completely burning, non-toxic and eco-friendly fuel commonly produced from lipids of plants and microalgae. Biodiesel production from microalgae has gained extensive interest in the recent year due to the presence of rich amount of saturated and unsaturated fatty acids such as palmitic acid, stearic acid, linoleic acid, etc., which are essential for production of high quality biodiesel. In the review of literature, there is no reports are available on this biodiesel property of Dictyosterium ehrenbergianum. Hence, in the present, investigation were undertaken to evaluate lipid production, fatty acid profile, and biodiesel property analysis of Dictyosterium ehrenbergianum. Coming to the material method, we have selected 14 different freshwater samples for isolation of these microalgae, and we are used boards basal media and as well as BG11 agar media for isolation of algae from freshwater sample. The algae, that is Dictyosterium ehrenbergianum, were identified using PCR-based 18S RDNA sequence analysis. Here, the lipid extracted from this microalgae were estimated using gravimetric method followed by fluorescence spectrophotometric analysis. Here, the lipid isolated from this microalgae were chemically characterized using GCMS analysis. The PEM product obtained from this microalgae were subjected for biodiesel property analysis, particularly kinetic viscosity estimation, flash point estimation, total acid number estimation, density, and acid content estimation. This based on 18S RDNA sequence analysis, coming to result and result. Based on 18S RDNA sequence analysis of 5TS gene isolated from isolate 5, that is Dictyosterium ehrenbergianum, were identified using NCI gene bank. This is a genome sequence of this microalgae. This is the accessory number obtained from NCBI gene bank. Coming to this is under microscopic image of Dictyosterium. This is the first one. Second one, fluorescent microscopic image of this Dictyosterium. Here, we are isolated more than 20 different algae. Among 20 different algae, we are selected 6 isolates, that is isolate 1, 4, 5, 8, 11, and 14. Among these different isolate, that isolate 5, which is identified as Dictyosterium erenbergianum, it containing high amount of lipid. The approximate lipid content is around 38%. Here, different physicochemical parameters on biomass and lipid production of D. erenbergianum were estimated. The highest amount of lipid as well as growth for optime at temperature 28 degrees Celsius, temperature ranged between 28 to 30 degrees, pH ranged between 80 to 9, and NaCl concentration ranged between 0.5 to 0.2 mg per liter, and maltose and sucrose served as carbon source, and ammonium nitrate served as nitrogen source. In this concentration, we get maximum amount of lipid as well as biomass.
Sorry to interrupt,
Coming
sir.
to this analysis of hello. This is a FAME product, GCMS.
Excuse me, sir.
Sir.
Yeah, yours 5 minutes over. Kindly within 1 minute. Okay.
Okay. This is the GCMS spectra of this FAME product of D. erenbergianum. Here we are identified oleic acid and palmitic acid as a major fatty acid. This biodiesel property of this organism, the flash point is 0.36, and also kinetic viscosity is 3.56, flash point 103, density 0.86, and hash content 0.01%. This value is within the comes under our value compare with the standard values obtained by India and American standards. This value comes within the limits. Yes, we come to conclude that we can exploit D. erenbergianum as alternative agent for production of biofuel. Thank you, sir.
Okay. Yeah, thank you, sir, for your presentation.
Okay.
So, from audience, anybody have any questions?
Sir, I'm Dr. Ravi Kiran. Can I present? Hello.
Can you hear us?
Yeah.
Hello.
Yeah. So you have identified alternative biodiesel, right? So
Yes.
yeah. So just where it would be useful or its utility approach, practical utility approach, where it can be used.
Madam, as per our knowledge, madam, we are isolated the biodiesel from this Dictyosterium. This value is approximately compared with the Indian standard as well as American standard. The range within the comes under the standard value obtained from America as well as Indian values. That's why we can exploit in large scale production, madam.
Okay. Thank you.
Okay, thank you, sir, for your presentation. So you may stop sharing your screen. So you can stop sharing your screen from your Zoom app.
Okay.
Yeah. So Dr. T.S. Harsa, so are you
Sir.
ready?
Yes, sir. I am ready, sir. Can you allow me to present my PPT, sir?
Just a minute, sir. So yeah, now you can proceed.
Yes, sir.
So Dr. Harsa, you have 5 minutes to present.
Yes, sir.
Okay.
Yes, sir. Thank you, sir.
Yeah, you can start now. Yeah, you can start.
Good morning, everybody. I have took this is not a research paper, that's a review article. This is on, everybody knows that this is a ozone conservation, past, present and future, how it was there and what is present situation and how it will be there. So welcome to all the delegates, awardees and participants of organizers and National Environment Science Academy team. So everybody knows that the ozone is one of the fragile shield gas which is going to protecting us from the ultraviolet radiations. And at the same time, it is a triatomic gas. Everybody, that's a simple concept. Triatomic, colorless gas, and chemically, it's a very active. Whenever it reacts with the other components like a chloride, so immediately it will going to dissolve, and this is what the major happening because of this chloride atom or a chlorine atom, that's ozone is going to depleting. And as per the director of UN Environment Program, so he has stated that the first truly global treaty that offers the protection to the every single human being is that ozone treaty. It may be a Montreal Protocol or a Vienna Convention. So everybody knows that this is ozone, and everybody is thinking about what is the ozone hole, a depletion of the ozone lower at the stratosphere. So only thing is we even present situation, the ozone is also present in the atmospheric region, which is considered as one of the greenhouse gas. Whereas we are all thinking, or we are all worried about the depletion of ozone, particularly in the stratosphere. So this story will going to talk about the theory that the ozone layer is a stratosphere might be damaged by the human intervention, preceded to the actual observation of the ozone hole because of the freons, which is used by the refrigeration as well as the AC, and also various types of liquid solvents which are using at the household level or household appliances. This is what which shows about the ozone layer, which is at the stratospheric zone, and it's starting to decrease and creating a hole at the Antarctic and Arctic place. And this is produced naturally photochemical reaction in the stratospheric ozone layer, which is called as a good ozone. Now we are talking about the good ozone, which is decreasing. Whereas it's another one is there. It is there in the presence of atmospheric or tropospheric zone, which is called as a bad ozone, which is also regarded as a greenhouse gas. Whereas this is what the peak which has been, there is a two peaks are there, like in the altitude of stratospheric, this is the one which is decreasing nearly, and also increasing. Another peak is at the in the tropospheric zone, where it is depleting, and it is increasing because of the various types of human activity. And as per the Chapman mechanism balance is being disrupted by the introduction of a chlorofluorocarbon and other similar gases into the stratosphere. And everybody knows that the free chlorine atom, which is an active, which is going to destroying the ozone hole, which is created a ozone hole at the Arctic and Antarctic ozone area. And this is a various types of gases, which is earlier we as a human beings we have not used this the solvents and the freon 11 and freon 12 because of the refrigeration AC which are using, and especially the single chlorine atom which is going to be destroying hundreds and thousands of the ozone molecule during its residence in the stratospheric. And this this theory was proposed in 1974, but due to the supportive document he could not able to propose, but later they have got know that it's this ozone layer is happening, it's because it really, and they have got the Nobel Prize for this theory. And this is the one where ground-based ozone measure 56, the British survey team, and actually presently we are depending upon the terms like a total ozone mapping spectrometer that we are analyzing. So how much of the ozone ozone hole is creating every day because of the human activity. So Dr. Susan Solomon, she is one of the the scientists who devised a theory that correctly explained about the destruction of ozone by the chlorine compound in the first time 1986. So why it's only in the Antarctic region? Everybody know that this is a major the question. That's particularly it acts on regionality and seasonality, because it is the most severe at the Antarctic in the southern hemisphere, and during the particularly in the spring season like September and October, whereas it's a less depletion which is in the Arctic, particularly in the northern hemisphere in the February and March, because the seasonality varies from place to place. So exactly the why it is Antarctic and Arctic region means there is a two important major the reactions will take place. One is a unique circumpolar circulation pattern over Antarctic in the winter, which isolates the stratosphere inside a vortex and acts like a containment vessel in which the chemical reaction may occur near the isolation. This is what created the splitting of the O3 into O2 and the chlorine atom that will going to attach. Another major aspect is the presence of polar stratospheric ice clouds. That is also major the environment which is supporting to the decreasing in the ozone content. So this is what you can see that's in with the diagram. This is the polar stratospheric cloud over. Yes, sir.
Yeah. So you have 1 more minute left. Kindly quick.
So these are the whatever the so far happened. What we can do is in the present situation, how we can conserve, how we can preserve, and what will be our role means. So whenever the automobile went on, so we have to put off the automobile or a vehicle which is using, and carpooling, use the mass transit and walk, bicycle, or reduce driving, especially in the hot summer days, and be careful not to spill gasoline while filling the cars like in the gas or any petroleum products. Also make sure that your car tires are properly inflated and maintained very well, and volatile organic carbon like that. And major thing is what we have to do is there is a chlorine and a bromine which is present in the stratospheric that we have to reduce according to the present situation. So the monetary protocol, which is called as a monetary protocol, is under Kigali amendment, which is shown that it's by acting together anything is possible. Because and recently in the pandemic situation we have observed that a lot of the ozone is recovering. And everybody even published also that is a research because of that. But only this thing, the climate change, and so let us act now to slow climate change, feed the world hungry, and protect the planet that we all depend upon. By working together, we can build a safer, healthier, most prosperous, and resilient world for the people who protecting our planet, which is because we are only one. We have the home that is called as Earth. Thank you very much.
So, thank you, sir, for your presentation. So from audience, anybody have any questions? Dr. Harsa, can you hear us?
Yes, sir. Yes, sir. I can hear, sir.
Yeah. So from expert, so you have some questions. Okay.
Yes, sir. Please.
I don't have a question. I just want you to make a point clear that you mentioned in the slide that near the polar regions in Antarctica,
Yes,
you mentioned about some chemical reactions occurring, and they are the major zone of ozone depletion.
Yes, madam. Yes, madam.
So, are they natural phenomenon, or is it has something to do with human activities? So
no. Actually, the 75% is a natural phenomena, and the remaining is 25% is because of the human activity also, madam. Because of the seasonality, what I have told, it's a seasonality and as well as the regionality that plays an important role, because southern hemisphere and northern hemisphere, the various the seasonality that is affecting, and also the increasing the circumference reaction which is taken place at the particularly in Arctic and Antarctic region when compared to other places like in India and other world global level.
So that is the atmospheric reaction.
Yes, madam. Yes, madam. And another human contribution is because of the using of these aerosols. That's particularly which is used in the aeroplanes and jet air, the rockets and all those things. That is a 25% which is contributing in the Arctic and Antarctic region.
Dr. Harsha.
Yes, ma'am.
It was a great presentation. I mean, I just want to ask you that have you published a review of this?
No, madam. Actually, I have sent it for the publication, or not yet get any reply from this, madam.
Okay. And what I understood from your presentation is
Yes,
that
madam.
CFC not.
Yes, chlorofluorocarbon, madam.
Yeah, chlorofluorocarbon. It does not shows any impact on ozone layer, but chlorine, right? So just I have one question, like in dengue or we frequently do chlorinated water, right, in
Yes.
our tanks and all. So
Yes, madam.
does it affect our atmospheric ozone too?
No, madam. That's not going to affecting anything on the ozone. So particularly the chlorinated water, that's not a very less concentration we have, madam. Whereas the chlorine atom, what we are telling is the chlorofluorocarbon and also the present we have changed our technology into the hydrofluorocarbon. So there are a lot of other substances which has this reaction which has been taken, CFC 11, CFC 113, and there is so on. There is a CSV 12 and 114. Like by class 1 and class 2, two type of chemicals are there. Class 1 is a chlorofluorocarbon and class 2 is called as a hydrofluorocarbon. Apart from that, so bromine and brominated carbon also there. That's major is the chlorine, which is very active. That's what you are telling is the chlorine which is present in the water or chlorination, that will not going to be much active. So whichever is the active chlorine is there, that will going to be react with this ozone, and that is depleting, madam.
Okay. So I'll just suggest just publish this.
Yes, madam. Definitely, madam. Definitely, madam. I will publish it, madam. Thank you so much, madam. Thank you so much for your work.
Okay. And Dr. TS, sir, thank you so much for your time.
Thank you, sir. Thank you so much, sir. Thank you, sir.
Okay. So may I request Mr. Ravi Kiran T for the next presentation? Are you ready? Mr. Ravi Kiran, are you there?
Sir, I am ready. Can I share screen? This.
Yeah. Just a minute. Yes. Now you can share your screen. So you have 5 minutes to present.
Yeah. Can I start, sir?
Yeah. You can start.
Good morning. Today I am just presenting the review article.
Kindly just put it on full screen mode.
Yeah, it is in full screen only, sir.
Yeah. So slideshow mode, you can put it. Yeah.
It is in yeah.
Okay. You can go ahead with this.
So, good morning. My name is Dr. Ravi Kiran from Department of Biotechnology, Bangalore University. So, this is the topic that is the nanotechnology in sustainable development. Today, how nanotechnology is used in sustainable development. This is the overall review of the talk. So, the world is facing great challenges to meet the basic commodities as well as finished goods and services. Today, we are facing the challenges in these areas. So the main aim of sustainable development to minimize the impact of these human activities on the climate as well as the environment. So the National Nanotechnology Initiative, which is taken place in 1997, they proposed that nanotechnology could be a more sustainable solution for all these global challenges. The main research agenda of this nanotechnology is on key issues, whether we can improve the sustainability in energy, water, food, shelter, transportation, healthcare, and employment, or we can develop a sustainable manner that with maximum benefits and we can minimum impact on environment as the climate.
Sorry to interrupt, sir. Your slides are not moving.
Yeah, only one slide I am doing now. I am moving the second slide.
Yeah, but from in our side, it is still in first slide.
I am already in second slide, sir.
So can you please once again share it? Just stop sharing and once again put it on. Yeah.
I will again share.
Yeah. Now you can share once again.
Post disable participant screen sharing. It is coming. You have to enable it, sir, screen sharing. Now it is visible. Second slide.
Yes.
Yeah. So these are the key properties of the nanomaterials, what we develop for sustainable technologies. All these nanomaterials, they have selective targeting, they have an increased affinity, they can improve catalytic and magnetic properties, and they have superior electronic and optical properties, and they have large surface areas, and we can chemically modify. All these properties the nanomaterials has, and these are the some of the nanomaterials that we can build our next generation of sustainable technologies. Already we are using a carbon nanotubes, graphene, fullerenes, zeolite, hyperbranched polymers, as well as dendrimers. And these are some of the metallic nanoparticles we are routinely using for so many types of biological applications like iron, silica, copper, zinc, cerium. All these nanoparticles, we are actually working on cerium oxide nanoparticles also. So these are the overview of the recent progress of these nanotechnology to overcome global challenges. One is in the area of water purification, clean energy technology, greenhouse gas management and material supply and utilization, and green manufacturing and chemistry. Coming to water purification, we have availability of the clean water. So the water is the most critical problem that we are facing in the present day. And nanotechnology has made advance in the water purification. Today, we are everyone using reverse osmosis, nanofiltration, ultrafiltration, microfiltration. And today, we also have a pressure-driven membrane process for water desalination, and 3D micro nanofluidic systems are also there for chemical separation, analysis, sensing, and energy conservation. Coming to clean energy technologies, today we are most of the nanotechnology we develop on the clean and renewable energy technologies. Today we are using the solar power, where we are using hydrophotovoltaic electrolytic system, which utilizes the semiconductor nanoparticles that are coated on the metal substrates as electrodes for the generation of hydrogen coupled with oxidation of these organic compounds in wastewater. Today we are all using, and we are using the membrane fuel, polymeric electrolytic membrane fuel cells for energy conservation devices for automotive applications. Next is about greenhouse gases management. Today, last speaker also told about the ozone, all these things. Today, nanotechnology is mainly useful for carbon dioxide separation, capture, and storage. Thermally rearranged polymer membranes with nano engineering cavities, they are tuned for carbon dioxide separation. We are also using nanostructured materials with enhanced electronic, optical, magnetic for high capacity carbon dioxide separation, capture, as well as storage. We also use zeolithic imidrolic frameworks for selective carbon dioxide capture as well as storage media. And next is our material supply. Here we are using most of the nanotechnology. Current problems for renewable energy technologies could be addressed by the following. So we can use nanostructured materials with enhanced optical, magnetic, catalytic property. We can convert e-waste also into mesoporous material that can help in the waste disposable problems, particularly with electronic products. And we can also use silica nanoparticles for the storage and controlled release of pesticides. And the last one, what we are today, is about green manufacturing and chemistry. As a biotechnologist, we are working in this area. So nanotechnology, green chemistry is particularly helpful in chemical, petrochemical, pharmaceutical, as well as material processing, all of energies. As I told you, we are working on developing the metallic nanoparticles using bacteria and fungi as a plants, compared to the physical and chemical methods where we have to use lot of toxic capping and destabilizing agents. So with the help of these bacteria and fungi and plants, we are working on green chemistry, that is synthesis of these nanoparticles using the extracts of fungi or the plant, or most of the metallic oxide nanoparticles are synthesized by using this green chemistry. So the main conclude my topic as the global sustainability challenges that world are complex and involve interdependent areas. And nanotechnology today it is a very versatile platform for the development of technical solutions to global sustainability challenges that are facing the world. So the novel nanomaterials such as water soluble, supramolecular fibers, membranes have emerged the promising blocks for a sustainable applications. So we have to take into consideration before the large development of nanotechnology based solutions and products for the global sustainability challenges that are facing the world. So we have to address all this issue before using the nanotechnology based solutions. So thank you for giving me an opportunity. It's a small topic, a review topic just I have put for my presentation. Thank you.
Thank you, Mr. Ravi Kiran. So from audience, anybody have any questions? Okay. So some questions from our expert. Okay.
Morning, Dr. Ravi Kiran.
Yeah, tell me.
I have a question. As you mentioned that nanotechnology can be used for the water treatment.
Yeah, water treatment also we can use.
So I have a question. Like, is it any process or is it any method that nanoparticles can be used to disinfect the biological wastes, like the bacterial load?
No, we are working on that, no, madam. Nanoparticles we are using for biological waste treatment also. We are using nanoparticles. Coating we can do the doping method, and we are using. Still the work is an infant stage. They are working on that. Particularly water treatment, already a lot of membranes have been developed based on nanotechnology.
Only limited to the wastewater treatment, or we can use the municipal water or the real water?
Now at present they are using for wastewater treatment also they are using. It is not like that they are using, but still it is not much work has been done now.
Yeah, Dr. Ravi Kiran, your presentation.
Hello.
Can you hear us?
Yeah.
Okay. So I have one question. I mean, nanotechnologies are emerging these days, but what I feel the major drawback is of cost. So would you like to comment on this?
Madam, cost means depending on the type of the material. If you synthesize the zinc or like a silver nanoparticles, if you are using, the cost of material will be high. But you are using a simple material like cerium oxide or a silica, the price is little bit less on synthesis of nanoparticles, particularly if you are going green chemistry. By using a plant or a fungal extract, that is easy to produce in the lab compared to the chemical methods.
Okay. Thank you.
Okay. Thank you, Dr. Ravi Kiran, for your presentation.
Thank you, sir. Thank you.
We will take next Mr. Yufana Riaz. So are you ready?
Yes, sir, I'm ready.
Okay. So meanwhile, just a minute. Miss Pratibha Tripathi, we will just take you up next after Ufana Riaz. Okay. So please be ready with your presentation.
Okay, sir.
Okay. So Mr. Ufana Riaz, yeah, please share your screen. Kindly put it on full screen mode.
Is it visible now?
Yeah, it's visible. So put it on slideshow mode. Okay. You can start now.
Very good morning to all of you. My work is on designing of photocatalytic materials for their application in wastewater remediation. So in this system, we have used polyaniline coupled tin oxide system for the degradation of an anti-inflammatory drug under normal solar light. So it's basically a method in which we are using heterogeneous photocatalysis, and as visible light is photocatalytic mechanism is regarded as a green approach, we thought of improving the efficiency of the metal oxides by formulation of their composites with polymers which can act as sensitizers. So we have basically used tin oxide, and we have used polyorthophenylene diamine which was chosen as the polymeric material. It's an electroactive conducting polymer, and it has a narrow band gap, and it shows good, in fact, outstanding electrochemical and optoelectronic properties. And this combination was not used earlier, so we have developed this system. And we have characterized the materials using the normal techniques, which are IR, UV, and XRD. This was the mechanism that was adopted. We have used a simple ultrasound-assisted technique to polymerize the monomer in presence of the metal oxides and to formulate their nanohybrids. So we have confirmed first the loading of the polymer using the thermal analysis technique, and we have confirmed the loading to be as much as we had taken, that is 24% of polymer in tin oxide, 16%, 12%, 18%, and 6%. And this was confirmed by the thermal analysis. Then we have done the morphological studies along with the RAX measurements to see the content of the tin oxide in the system, and it was found to be in the stoichiometric ratio as we had theoretically proposed. And we have obtained different structures in the morphological studies, which shows that these systems are showing different organized morphologies depending on the loading of the polymer. So the polymer in fact tune their morphology from spherical particles to rod-like structures as we increase the loading. And then we have done the contrast imaging that could show the content of tin and the content of the polymeric components, that is the presence of nitrogen and polyorthophenylene diamine, how much it was covering the system. So by this imaging technique, we could actually see how much surface was covered by the polymer or encapsulated by the polymer. These were the XPS measurements to see the state of the conducting polymer and the oxidation state of the tin oxide as well. And we could see that there is a strong electrostatic interaction of the tin oxide system with the polymer. And these were the X-ray measurements, which showed that the crystallinity was quite intact even upon loading of the polymer up to 24%. So the crystalline state of tin oxide was in fact unaffected by the presence of the conducting polymer. That means it was found to somewhere encapsulate the structure and cover the entire area of the tin oxide nanoparticles. Then we even did the TEM analysis, which shows agglomerates of these spherical particles, which were as small as 16 to 21 nanometer. They covered a very small range. And the IR spectrum was intentionally done to see the electrostatic interaction of the NH group of the present in the conducting polymer with the oxygen of the tin oxide. And we could see that there was an intense interaction which held over the surface of the tin oxide. And then we did the DRS measurements to see the improvement in the band gap. So we could see that the band gap was effectively reduced when we were loading the conducting polymer, and therefore it was applicable to be used in the visible light degradation of the concerned drug. And these are the photocatalytic activity experiments, and we could see that there was approximately 92% degradation of brufen drug that we had chosen as an anti-inflammatory drug within 60 minutes under the normal solar light. So we did the kinetics, and we've seen that the system was quite efficient in degrading the drug within a period of 1 hour. And recyclability test measurements were carried out to see whether the catalyst was affected after how many cycles it could be used. So we could see that up to 5th cycle, we could obtain recovery of the catalytic material. So it was found to be quite stable. And then did these scavenging studies to just the generation of the radicals that were responsible for the degradation of the drugs. And this shows that the production of hydroxyl radicals was maximum, which could be the radicals that are responsible for the degradation of the drug. And we did the LCMS studies to see the fragments that we had obtained, and we could see that the fragments that were obtained, the lowest byproduct, which was around 81 and 82 of molar mass 81 and 82, which was found to be completely non-toxic. So this was the catch of the entire work, that we could degrade an anti-inflammatory drugs into particles that could be or products of molar mass fragments that were potentially non-toxic in nature, because usually the byproducts that are produced are potentially harmful than the parent molecule. But in this case, we could obtain products that were potentially less toxic. And then we did the comparative study of the work that has been carried out based on tin oxide, and we could see that we achieved maximum degradation in visible light, which was not reported by the other authors. And therefore, this work was significant in the terms that the use of conducting polymer coupled with tin oxide could reduce the degradation time, as well as it could show maximum activity under solar light radiation. So this work was published in Journal of Materials Research and Technology, and it has been widely cited.
Thank you so much for your presentation, Ms. Fana Rias. So any questions from our audience?
Good morning, Dr. Ufana.
Yeah, good morning.
I have a question. So what was the time of sunlight exposure in your experiment?
Time of sunlight exposure, we have taken the maximum time to be 1 hour.
Okay.
And we actually carried out the experiment at up to 2 hours, but we could obtain maximum degradation within 1 hour. So we've reported all the results within an hour, that is 60 minutes.
It was natural sunlight, or you used any light source for this?
Your voice is very low. Can you speak a little louder?
It was natural sunlight, or you used any light source?
We've used normal solar light, which is obtained during summers.
Okay.
Okay, thank you, Dr. Rufana. So you can stop share your screen. So Dr. Pratibha, so are you ready with your presentation?
Yes, sir.
Yes, ma'am. You can share it now.
Yes, I'm sharing. Download.
So meanwhile, Mr. Mohammed Hassan will review with your presentation. We'll just take you next after Dr. Pratibha Tripathi.
Sir, it is visible?
No, ma'am. It's not visible.
Not visible. Okay.
Ma'am, are you facing any problem?
Yes, sir.
Till now, ma'am, it's not visible, your presentation.
So now is it visible?
Yeah. Okay. And now you can start.
Okay. Very good morning to all of you. This is Dr. Pratibha Tripathi from CSIR-Institute. I'm working as a ICMR RA with Dr. Pooja Khare ma'am. Today the title of my work is Amelioration of Atrazine-Induced Toxicity by Endophytic Bacterium Bacillus species strain CMAP-A7 in Andrographis paniculata. Now, what is atrazine? Atrazine is among one of the most common herbicide and ranks second due to its extensive global utilization, and it is being used for protecting crops from unwanted animal grasses and broadleaf weeds. And due to its intensive utilization, atrazine and its residues in soil has become a serious threat to the mankind and to the ecosystem. It has been found to be readily absorbed and as accumulated at high concentration by plants and affect crop production. Due to its toxicity on non-target organism, it is considered as a serious threat. The mode of atrazine action in plant is as it hampers the photosystem too, and hence hamper the chlorophyll synthesis and the photosynthesis process. And it also produce the reactive oxygen species and produce the oxidative stress in plant, which hampers the normal plant growth. Hence, the protection of plants with this hazardous component is very crucial. Now, why I have selected the medicinal plant for my study? The pesticides are often used to improve the crop yield and profit margins in the production of medicinal plants, and the pesticide residues in medicinal plant is of safety concern to its consumers. Hence, the accumulation of pesticide more than the recommended dose of by IARC and FSSAI is a serious issue to check out. The model plant for my study is Andrographis paniculata, which is a herbaceous medicinal plant, and it is known for its bitter taste and also known as Kalmate. It's high due to its high therapeutic properties and therapeutic and curative properties. It is used for the treatment of several diseases such as fever, respiratory inflammation, insect bite, etc. And in recent years, it has been observed that it is found effective in binding the reactive site of SARS-CoV-2. So it is also has become a main plant in demand. What are endophytes? In recent days, to check the problem of pesticide and herbicide accumulation in the plants, the various mitigation strategies has been employed, but the most strategies are either expensive or not feasible check out. So to reduce the accumulation of such hazardous toxicant in the plant, the application of microorganism and especially the endophytes is of really important, because endophytes are the organisms, often fungi and bacteria, that live between the living plant cell, and application of these endophytes in the plant system has been observed that it promotes the plant growth promotion by imparting several mechanisms, like it stimulates the accumulation of crucial mineral nutrients, phosphostabilization, siderophore production, etc. So in case of if a plant accumulate a toxicant like atrazine, the endophyte hamper the accumulation of the toxicant in the plant, also reduce the phytotoxicity, and also reduce the oxidative stress. Then this is the schematic representation of endophytes for atrazine in reducing the atrazine-induced toxicity in the plant system. These are the objectives of my study: the isolation of endophytes from the atrazine-contaminated sites, and then in vitro screening for their atrazine degradation potential, and assessment of plant growth promotion rate. And also a greenhouse test has been conducted to review the potential of the selected endophyte in imparting stress tolerance as well as with reduced atrazine accumulation in the plant system. For this issue, I have selected the 2 doses for my study, that is the 25 ppm and the 50 ppm. These are the material methods. Now, these are the results of my study. In this study, I have isolated the endophytes from the plant system of sugarcane and Oryza sativa from the Barabanki and Lucknow agriculture fields of Uttar Pradesh, which are known for the atrazine contamination. The number of microbes isolated were the 37 bacterial and 15 fungal strains. The microbe selected is the CMAP A7, and its physiological and molecular characterization has been shown, and on the basis of that, it has been considered as a Bacillus ghemocensis on the basis of its 16S RNA sequences, and NCBI, and the NCBI accession number has been already shown here, and it is on the physiological basis, it is the potential indolactic acid producer, phosphorylation, CYP4 production, SSC deaminase activity. We have selected this strain, and in vitro study, it has been observed that this strain successfully deplete the atrazine in the broth assay after 10 days. It means after 10 days, complete almost 99% of removal of the atrazine was observed in the in vitro assay.
Sorry to interrupt, ma'am. You have 1 more minute left. Kindly wrap up quickly.
Okay, these are the plant test results. In this, we are showing that the selected strain produces the plant growth, impart the plant growth promotion, and as well as it degrades the atrazine in the soil as well, which is shown as the residual soil atrazine content, and it also produces the atrazine metabolites, which are the less toxic than the parent chemical. This is the atrazine content in the plant tissue as well, and in this result, this is observed that the inoculation of this strain imparts the plant growth promotion as well as it limit the atrazine accumulation in the plant tissues, and it also reduce the soil atrazine content. And the secondary metabolite content, as I am working on the Andrographis paniculator, hence the medicinal value of this plant is due to the andrographolite content, and that content is about 2-fold enhanced in presence of the inoculated strain, and it is not affected in presence of the atrazine stress. This is the summary and conclusion of my work, that the inoculation of CMAP A7 imparts plant growth promotion with reduced atrazine accumulation in plant tissue, and this is the first study of atrazine mineralization by endophytic bacteria in the plant soil system, and this approach is potential in imparting growth of non-targeted atrazine resistance in plant, and the direct role of endophyte CMAP A7 in enhancing atrazine tolerance by inducing the host stress responses. Hence, the resultant is the safer produce and improved crop yield. This work of mine has been published in the Science of the Total Environment in 2021. Thank you.
So, thank you, Dr. Pratibha, for your presentation. So, anybody from audience, any questions?
So my question is, I mean, you have, I think, evaluated that, yeah, there is a reduction in the atrazine-induced toxicity, right?
Yes, sir.
Parameters. I mean, what parameters did you evaluate?
Sir, ma'am, your voice is very not very clear. Hello.
Now, am I audible?
Ma'am, your voice is very slow. It's very low. Yes,
Yeah,
yes.
Now, can you, am I audible now?
Yes, ma'am. Yes, ma'am. Yes, ma'am.
My question is, what were the toxicity parameters? I mean, what were the parameters you considered it as a toxic parameters, and you evaluated a reduction in this parameter means there is a reduction in the toxicity. So what were
Ma'am,
the parameters you selected?
ma'am, the parameters we have selected is the degradation in the plant bed and
Okay,
the atrazine. Yes, ma'am. And I have selected the
fine.
2 doses. In first dose, it is not imparting as such the toxicity, but in the second dose, it is imparting very much of toxicity, which is visible in form of oxidative stress. And atrazine accumulation of atrazine content is more important in our study because it is a medicinal plant and whole medicinal plant body is used in the medicinal preparation. That's why to check this problem, we have selected the remediation this method.
Okay.
Yes, sir. Yes, ma'am.
Okay, thank you, Dr. Pratibha. So you may stop sharing screen.
Okay. Thank you, sir.
Yeah. So, Dr. Mohammad Hassan, so are you ready? Dr. Mohammad Hassan, are you there in the call?
Yes, sir. Yes, sir.
Yes, sir. So, before starting, sir, I guess you have two presentation, right? Hello.
Yes, sir.
Yeah, one is the application of biomass waste derived biochars for removal of anionic dye from water.
Actually, this is my presentation. My presentation is the utilization for
Sorry.
the sustainable development. Biochar, the production of biochar, the way of utilization in sustainable development.
Okay, okay. So you will be presenting one by one, right?
Yes, sir. Yes, sir.
Okay, okay, okay. So kindly start with your first presentation, like application of biomass waste. Okay.
Yes, yes, sir. Yes, sir.
So meanwhile, Dr. Supriti Khan, please be ready with your presentation.
Yes, sir.
You will be next after Mohammad Hassan.
Okay, sir.
Mohammad Hassan, kindly share your screen. Okay, put it on slideshow mode. Okay, okay. Yeah, yeah. You can start.
Good morning, all of you. My name is Mohammad Hassan, working as a project, research scholar under the supervision of Dr. Pooja Khare. Today, I am here to present my oral presentation. And my oral presentation title is the biochar production, the way of utilization of agriculture waste system for the sustainable development. Before going to my presentation, I would like to share some related outpoint to which one taken from our study. According to the Ministry of Renewable and Energy, India generate on average 500 million tons of crop residue per year, which 92 metric ton is burned each year. A huge volume of these residue is burned on the farm to the clear for the sowing of next crop due to the shortage of human labor, high cost of removing the crop residue by the conventional method. Generally, crop residue of different crop contain 80% nitrogen, 50% phosphorus, and 20% potassium, which can replace the 6.5 million tons chemical fertilizer. Biochar is the solid carbon-rich product obtained from the thermal chemical conservation of biomass under limited or no oxygen environment.
And the biochar production from crop residue not only the solve pollution problem, but also provide the better input to the crop production and farmers' profitability. In my whole study, I have taken two objectives. The first objective is the biochar production from the crop residue and its utilization as the soil fertility, enhance and alternative fuel as a sustainable solution. The second objective is technological development of biochar production at farmer level are needed to discontinue the crop residue burning in India. So this is my material method. So generally biochar is produced by the thermochemical conservation of biomass under oxygen efficient condition through the slow pyrolysis, fast pyrolysis, and gasification. Slow pyrolysis is the conventional type of method that characterized by soil slow heating rate and long residence time. In this technique, the temperature is 300 to 500, and the biochar is 35 to 50%. And the second type of pyrolysis method is the fast pyrolysis, and the pyrolysis temperature is 450 to 850 centigrade, and the biochar is 10 to 12%. And last one is the gasification, and the pyrolysis temperature is higher than 800, and the biochar is 8 to 10%. The current status of crop residue production, burning, and emission in India. A huge volume of crop residue produced in India in two seasons. That is, the one is the Kharif, which one June to October, and the second is the Rabi, which one November to March. The major contributor crop of like wheat, paddy, sugarcane, and maize, which one 63% of the total residue produced in India. Uttar Pradesh has produced 72 metric ton, followed by Punjab and Haryana. The total quantity of residue burned in India is 98.4 metric ton to 131.9 metric ton. Among the state, UP, Punjab, and Haryana contribute to 20, 21, and 10% of total crop residue burning of India, respectively. In the large amount of NPK losses during the burning of crop residue in India. This is slide showing the revenue generated from the different crop residue derived biochar, and the second is improvement in the crop residue production through the biochar application. Though due to the high heat values, biochar could be considered as the alternative source of the energy. The total revenue which can be generated by the use of biochar for electricity production were calculated by the multiplying the heat value of biochar with a unit cost of electricity. The electricity produced from the biochar, that is wheat residue, has the maximum provide revenue. The energy generated from the biochar generally require initial setup, which not easily acceptable for the farmer having a low area of cultivation. This technology at the community level by the rich and progressive farmer having the large scale of the cultivation with the good transport facilities could be beneficial. The trade-off between biochar improvement in the soil and biocoal suggests that the farmer application is the more valuable in terms of carbon prices. And the crop yield improvement varied from with soil and biochar type, also application rate and the crop cultivate in the field. Biochar dosage and feed step could be optimized with the soil type get to the optimal result. There is a huge variation in the application rate 0.1 to 9.6 10 per hectare, and the enhancement in the crop yield is the 5 to 178% as compared to the control in worldwide. This slide showing the conversion of crop residue burning into the biochar. So in the November, after the harvesting of the rice, farmer have to practice burn the paddy crop residue to clear their field for the next succeeding crop. So biochar production via slow pyrolysis is a better option for the farmer for the mitigation of these their crop residue into valuable product biochar, and farmers use this biochar in the form of organic fertilizer. And the fast pyrolysis can give a loss of the emission, nutrient losing, and soil fertility. Emission causes the climate change and also health hazards. A biochar product Production biochar is the have say CO2 natural fuel or used as a soil amendment, which can reduce the emission and carbon and nutrient sequestration, will enhance the soil fertility and also mitigate the climate change and enhance productivity. So biochar incorporation in soil has the many parameters depend upon biochar properties. Biochar has alkaline pH and water holding capacity that remove the liming effect in soil and retain moisture and improve microbial activity, and the high CEC cation exchange capacity and nutrient which can use as a nutrient increase in nutrient availability, sustainable supply for plant available nutrients. And the biochar also has the high surface area and porosity, which one useful for the habitat for the microbes, adsorptive properties, reduction in the contamination and mobilization. And biochar is the main source that has the property, which one is the carbon recalcitrant, which can atmospheric carbon capture into the in form of sequestration into the soil and enhance the productivity. And biochar has the also has the adsorptive property, which can mitigate the many environmental contamination from the soil. And this is my conclusion. The biochar production from crop residue has the beneficial effect on the environment as well as economic useful the farmers. The total value of the carbon sequestration from the biochar could be 9 to 14 million dollars per year. The replacement of chemical fertilizer by biochar could save the 5,016 to 17,014 through the sequestration the nutrients. So now the biochar production is the attractive way of the reducing the deleterious effect crop residue burning, along with the multiple beneficial on the environment and for the farmers. Thank you.
Yeah, thank you, Dr. Mohammed Hassan, for your presentation. So regarding this topic, anybody has any questions from audience?
Yeah. So, Dr. Mohammed Hassan, I have a question.
Yes, ma'am.
What is the biochar production cost?
Actually, ma'am, biochar production cost yet not decided, but in the running form we can optimize the actually the cost of biochar and use for for the farmers. So it is not decide for the.
Okay. So I mean, if you have conducted some pilot study, so do you have the cost estimate? I mean, if you have done pilot study in your lab, so what was the cost?
एक्चुअली मैम जो इसकी अभी कॉस्ट एक्चुअली ऐसा मैम कि अभी डिसाइड नहीं, बट ये है कि फार्मर्स के बेनिफिट के ऊपर हम लोग इसकी कॉस्ट डिसाइड करेंगे एक्चुअली जो फार्मर्स के यूजफुल हो, ताकि वो हम लोग जो प्रोड्यूस कर सकें या उनकी एक्चुअली हम लोग ट्रायल भी मैम करते हैं उनकी फील्ड पे जाके, तो वहाँ भी हम लोग डिसाइड करते हैं कि बायोचार कैसे, जैसे उन लोगों की डिस्टिलेशन यूनिट है तो उससे भी हम लोग बायोचार प्रोडक्शन बता सकते हैं कि आप लोग ऐसे बना सकते हैं, जो उनके यहाँ से रेजिड्यू क्रॉप जनरेट होते हैं इनकी फील्ड से, तो वो भी एक्चुअली बना सकते हैं। और अभी कॉस्ट मैम डिसाइड नहीं हुई, बट ये चल रहा है मैम, जो हमारी मैम सुपरविजन है वो एक्चुअली अभी डिसाइड नहीं की इसका।
Okay, so because whenever we are I mean representing the applications or related things, so we have to be clear ki what is the cost, right? So
okay, ma'am.
Work on it. Okay, what is the cost estimate for the biochar production? So it would be helpful to you.
Yes, ma'am. Yes, ma'am.
Do you have a question? Okay, fine. Okay, thank you, Dr. Hassan.
Welcome, ma'am.
Yeah, Dr. Hassan, can you proceed with your second presentation?
Okay, sir. Thanks. Your presentation is now.
Sorry?
एक्चुअली जो सेकंड प्रेजेंटेटर है सर, उनके पास एक्चुअली लिंक नहीं आई। मयंक सिंह हमारी लैब से हैं सर, तो अभी उनके पास कोई ऐसी लिंक नहीं है जो उससे वो अपनी शेयर स्क्रीन कर सकें। मैम वो सर वो शाम वाला जो प्रेजेंटेशन है उसके रिगार्डिंग लिंक है, लेकिन इस वाले में अभी उनके पास कोई ऐसी लिंक नहीं है जिससे वो
Okay. Wants to present. Fine. So we can proceed with the other presenting.
Yeah. So Dr. Essen, you may stop sharing your screen.
Okay, sir. Thank you.
Okay, thank you, sir. Dr. Supriti Khan, are you ready?
Yes, sir. Yes, sir.
Okay.
So will I share now?
Yeah. So just a minute, ma'am.
Good morning. This is Dr. Namita Das. May I request, please? Sir?
Yes. Tell me, ma'am.
Would you please permit me after Supriti Khan, please? Because some of the electricity related work that will be going on in my room. So I may not be able to present later on. So just a request.
Okay. So after Supriti Khan, we will take you up next, okay?
Thank you so much, sir. Thank you.
Gunjan, can you be after Namita Das, please?
So will I share?
Gunjan @Ritu, are you there? Okay. Supriti ma'am, you can start.
Okay, sir.
Okay, sir, I'm starting.
Yeah. Please share your screen.
Yes, sir. So is it visible?
No, not till yet. Are you facing any problem with sharing the screen?
Yes, sir.
So meanwhile, can you please fix it of your problem so we can take Dr. Namita Das?
Yes, sir.
Okay. So we will just take you up next, okay, after Dr. Namita Das. So Dr. Namita Das, are you ready with your presentation?
Yes, I'm just sharing my screen. So
Just a
I'm
minute.
disabled.
Yes, ma'am. Now I can share.
Okay. So my slides are visible, sir.
Yes.
So can I keep this in this form only, because in the PowerPoint mode, actually my slide won't be moving. There are some issues in my system.
Okay. You can go ahead with this.
Okay, so thank you so much. So basically, myself, Dr. Namita Das Saha, I am a scientist senior scale at Division of Environment Science, representing ICIR, Indian Agricultural Research Institute. And presently I am presenting from New Delhi. So today I'll be presenting one of the topic that is for suitable and very time, it is apt for this particular time which we are running through at Delhi. So presently we all know the air quality index of Delhi is has become very worsened. So at this moment the rice straw burning and all these issues are being described every day. And here I'll be presenting a very short finding which we did last year. So basically we tried to utilize rice straw for different alternative purposes, for their best alternative purposes. So basically we think about that rice straw is actually a waste, but we can have many more alternative valuable uses for these rice straws. And what actually we should do before going for prescribing different alternative uses of rice straw. So that attempt we have done here. So just to give you an introduction regarding the rice straw amount. So basically worldwide, we do have 731 million tons of huge amount of rice straw every year that gets produced worldwide. And for India, it is around 126.6 million ton. And it is also a huge amount. And the unfortunate part is that 60% of this valuable biomass is actually being burnt in the field. And not only it produces the different obnoxious gases in the air, but it chokes our that breathing system, and basically the delegates, Haryana and Punjab, we feel it very badly. And at present, you know, whenever we go outside from my campus, we feel like eyes are burning. So that's an issue presently we are feeling here. So basically this particular huge amount of rice straw biomass that can be alternatively used for different potential uses, like for bioethanol, biochar, mushroom cultivation, and many more. And by composition wise, rice straw is basically comprising cellulose, hemicellulose, lignin, and silicon. These are the main 4 components which is basically present in rice straw. And different type of varieties, they contain different amount of cellulose, different amount of lignin, different amount of silica, hemicellulose in them. And this is the point actually for which some of the varieties of rice straw that will be suitable for some one purpose, and some of the other varieties will be suitable for other purposes. So that's how these all varieties which are predominantly being cultivated in the northern area, before going for any prescribing for any alternative uses, one must go through the characterization of all those rice straws, different varietal rice straws. So here actually we did. So in northern India, although many rice varieties are predominantly being cultivated, but we identified the 17 most predominantly cultivated rice varieties. We have collected the rice straws. We did morphological features analysis through scanning electron microscopy. Biochemical components for cellulose, hemicellulose, lignin, silica were characterized in the lab, and functional group analysis were done through FTIR. It's fine. So basically, these are some of the representing pictures of different FTRs of our different varieties. And basically in this blue box, actually, we characterized different peaks for lignin, hemicellulose, cellulose by different corresponding peaks that I have presented over here. And also through scanning electron microscope, we have studied the presence or absence of silica groups, grains, surface roughnesses. So these are the parameters we have analyzed. So now this is some of the descriptive statistics of biochemical properties of our chosen varieties of northern India. So basically here you can see that Tarauri Basmati, it was containing your highest amount of lignin and also a good amount of your hemicellulose. Likewise, Pusha Basmati 1121 was containing highest amount of cellulose. So like that. So many varieties were totally, they were characterized. And here actually in the box itself, you can see that these were actually the desirable features of straw for prescribing them for different alternative uses. And based on those descriptive statistics, we have categorized of those varieties into high, medium, low category. Okay. So then based on these all properties, we have also studied through heat map, and particular clusters also appeared in their cluster dendrogram analysis. So ultimately the Venn diagram, here also we just group wise we have made them a club, that say for example like few of the varieties can be used for dual purposes, like Pusha Basmati 1121, Pusha rice hybrid 123, 122, Pusha 44 can be used for both of the purpose, like bioethanol, composting, like Pusha Sugandh 5 can be used for your composting and mushroom cultivation for the dual purposes. Likewise, Pusha 44 can be used for bioethanol and biochar purposes. So when we go for the best alternative uses, actually that will be giving you the best result for the best alternative purposes, and that will actually will encourage our farmers if we go for prescribing for those kind of uses. So that's how it was a small study here, and this year also in a wider way we are studying the same thing, and hopefully we will be going for some of the prescribing protocols for alternative uses of different rice varieties of northern India. So thank you so much. Thank you all, and hope we these alternative uses and all encouragement regarding this, and some of the policies are also being made through means it's in the process, so that will be encouraging for some of the alternative uses and also will be curbing down the burning processes. So thank you all. So if any question from anyone.
Okay, thank you, Dr. Namita. It was a good presentation. So any questions from audience?
Thank you.
And again, I do understand that in North India, this is a huge problem. But my question is, I mean, you have identified a couple of, varieties.
Ma'am, your voice is not clear.
Okay, so you have identified the best alternatives for a couple of varieties of rice, right?
Yes, yes.
Yeah, yeah. So my question is, I mean, this is, this is identified among the researchers. The researchers would read your paper, but actually, farmers are the one who are burning this straws, right? So
Yes.
the awareness regarding the alternative use must be disseminated among the farmers, right? So.
Yes.
So I feel that although you have done a very good research, I'll just ask you if your department or if your institute has NSS, and if they can do outreach programs to villages, and if they can create awareness regarding this alternative uses, then it will create an impact. Otherwise, it would be just in the research paper, and we, the researcher, would read, but again, the applicability would be, I think, will not be, fulfilled and all.
I appreciate your comment, and in this regard, I would like to let you know, please, see that high-level committee in the ICR. Basically, we had last 10 days back, we have a meeting along with some of the policymakers. So basically, these are some of the basic research which are being enforced by them, that we must identify, and also we must encourage those farmers actually for utilizing such alternative uses. Fine. So this is actually being going to be as a like policy or some of the awareness what you have talked about just now. So that are being in a process, and it's actually being happening, and it is actually the right time that ICIR, Indian Agricultural Research Institute, they have initiated this activity. And not only that, another activity parallelly of this particular program is being conducted for as Pusha composting. Okay, you might have already heard, Pusha composting, some of the microbial consortia, and it's doing havoc in the northern India, in Haryana belt, in Delhi. So another parallel program is also being going on. And this particular initiative we have initiated since last year, and we are in a process to make a policy and to enhance the awareness among the farmers also. So that's how. Thank you so much.
Okay, thank you.
Any more question, please?
Good morning, Dr. Namita. I have also related a question. Like you have described about the processes. I just want to ask that are these processes economical for the farmers that they choose these processes instead of burning their crop wastage?
They will be given the incentives for that. If suppose they choose for utilizing their straws for these alternative purposes from the government itself, we are planning in such a way that they should be given some of the incentives. So that's how. Otherwise, the economics aspects also, we are presently working on that. How economic will be there instead of burning and all these things. So that's a challenging task, but we are pursuing this not only as a research aspect, as madam said earlier, madam. So basically we are pursuing to have certain alternative prescription regarding out of this particular work. So that is how.
So you are saying that you people have not figured out the cost, and if it is not economical for the farmer.
Madam, I have already shared that this work we have already initiated only last 1 year, and we got another second rice season here, because in Delhi we get only one rice season from the Kharif only. That's it. Fine. So initial work which I am presenting, it is our last Kharif seasons, and this year again we are pursuing, because rice only just very recently harvested. So things are in a process in the laboratory, and all details are being worked out presently.
Also, Dr. Namita Das.
Yes, madam.
Very good.
Madam, your sound is not clear.
Again, I feel that your research work will have an impactful utility approach. Just plan it out, okay, how it can be disseminated among the farmers and how we can take it to the best.
Yes, madam. Thank you so much. Thank you.
Okay, thank you, Dr. Namita.
Thank you so much.
So, you may stop sharing your screen.
Yes.
So, Dr. Supriti, have you fixed your problem?
Sir, I'm telling that I'm sharing the screen when from sir. I have two accounts, Supriti Khan. So, sir, in one account it is showing that the host disabled screen sharing. So, can you?
Now you can share it.
Yes, sir. Thank you, sir. So, is it visible?
Yes, yes.
Okay, sir. Thank you, sir.
Put it on in.
Okay, sir. Good morning to our respected chairperson, co-chairperson, other committee members, and my dear colleagues. So today we have gathered here for an international conference, and the topic of my presentation for the conference is abatement of fluoride from polluted water and its ability to support sustainable development with possible policy suggestion. It's basically a review. So before starting, I would like to say that in the recent scenario, the contamination of the water resources by different ions is a major problem worldwide, and among which the contamination of the water resources by fluoride is a severe problem, which not only causes the pollution of the water resources, but also the ingestion of which causes different health hazards and diseases. So in this presentation, I would like to talk about how the contamination of the water resources by fluoride is affecting us, and the different techniques that has been developed for defluoridation, and most importantly a literature review on the works of the different scientists on defluoridation. So proceeding forward, so these are the contents of my presentation, which includes introduction, sources of fluoride, fluoride in groundwater, causes of fluoride, health effects, different fluoride doses and effects on human body, mitigation measures, literature review, conclusion, and references. So before proceeding forward, we should know where the fluoride is coming from. So fluoride is a naturally occurring compound. It's derived from fluorine, the most electronegative and the most reactive. The most importantly, it is the 13th most abundant element in the earth, and the natural abundance in the earth crust is 0.6 to 0.06 to 0.09, and mainly it occurs in the form of different chemical compounds. The most important part of the introduction is that the maximum permissible limit of fluoride in we should intake in our body is 1.5 milligram per liter, and that is according to WHO.
So your slides are not moving.
Sir,
yeah, your slides are not moving. Can you do one thing, like please stop sharing a screen and put it on slides.
Okay. Okay, sir.
Yeah, yeah, now it's moving.
So
Yeah,
I'm doing it like this.
yeah. Go ahead,
Okay,
please.
okay, sir. So the most important part of the introduction was that the maximum permissible limit of fluoride we should intake in our body is 1.5 milligram per liter, according to WHO. And the most important diseases that has been caused by this is dental and skeletal fluorosis, and also includes other diseases. Moving forward, what are the sources of fluoride? We should know where the fluoride is coming from. It includes two sources, that is natural and the anthropogenic sources. These are mainly as natural sources. What are natural sources? These are mainly associated with minerals of the geochemical nature, or we can say that the breaking of the rocks, which causes the contamination of the water. Anthropogenic sources, it generally includes the burning of coal, extraction of aluminum, and use of phosphate fertilizers, etc. Now, one of the most important part of my presentation, how the fluoride contamination is affecting the world, and next, how it is affecting India. So as we can see the map here, and let me tell first that in 29 countries, 200 million people are affected with fluoride contamination. As we can see in the map, the most affected part are the deep red color parts, which includes some parts of Africa, the Middle Eastern parts, some parts of South America, North America, some parts of Australia. Moving on to India, 203 districts in 20 states of the country are affected with fluoride contamination, and the most affected part are the Rajasthan, Gujarat, and Andhra Pradesh, which is also shown by the deep red colored parts. So now what's the cause of fluoride contamination? It includes aquifer material, volcanic ash, and fertilizers. Aquifer material, that is the breaking of the rocks, as I mentioned before, volcanic ash, when the ashes comes to the surface due to the rainfall, and this causes water contamination by fluoride, and the phosphate fertilizers, which are one of the major problems for fluoride contamination. Health effects. So as I mentioned before, that fluoride always does not have a negative impact, it also have a positive impact. So the maximum permissible limit, if we intake fluoride in that amount, it's helpful for our bones and teeth. But lower concentration, that is less than 0.5, it causes dental fluorosis, and above 1.5, it causes skeletal fluorosis and even death. As we can see in the picture, how the dental fluorosis is caused and how the skeletal fluorosis is caused. Dental fluorosis is mainly caused due to the ingestion of excessive fluoride during enamel formation, and skeletal fluorosis is generally caused due to the excessive accumulation of fluoride in the bones. Moving forward, what are the doses and the effects on human body? Here only I will tell that, as I mentioned, that it has both positive and negative impact. As you can all see that from 0.5 to 1, it protection against dental caries, and it takes care of bones and teeth. And as mentioned before, I have told about skeletal and dental fluorosis. So as the water resources are getting contaminated, there should be some measures for preventing the harmful effects, which generally called as the mitigation measures, which includes two methods, the in situ treatment method and the ex situ treatment method. In situ treatment method is nothing but the direct removal of fluoride from groundwater, and it also involves rainfall recharge. The most important method is the ex situ treatment method, which generally involves 4 methods, that is adsorption, membrane process, ion exchange process, and the precipitation method. But most important is the adsorption process, because it is cost effective, and also the retention of the fluoride ions is also possible at a low concentration, and the adsorbents are easily accessible for a wide range. And before moving on to see which adsorbent we should use, we should do the characterization of the adsorbents, which includes it, because the removal efficiency of fluoride generally depends on pH, contact time, initial fluoride concentration, and size of the adsorbents. And I will mention that the removal efficiency of the fluoride is inversely proportional to the initial fluoride the pH and the size of the adsorbents. So the 4 methods, as I mentioned before, adsorption, ion exchange, precipitation, and membrane process, both have advantages and disadvantages. And as you can all see that adsorption, the fluoride efficiency removal is the highest, but the disadvantage is that it's highly dependent on pH, and we should maintain the pH before moving forward. The ion exchange, it retains the taste and color, but due to the expensive cost of the ion exchange resins, it's difficult to move forward with this phenomenon. And also due to the low pH, it's impossible to use. And the precipitation method, due to the use of alarm dosing, the pH is very high, but the advantage was the use of the Nalgonda technique, but which eventually failed later. The membrane process is to some extent useful because the parameters.
Sorry to interrupt, Supriti ma'am.
Yes,
Your 5 minutes are over. Kindly wrap up within 1 minute.
Okay, sir. So the literature review, I will like to tell that starting with Kumari and co-workers, the adsorbent used by them was alumina and sulfuric acid by activation, and the fluoride removal was 96.72%. Wambu and co-workers, they used soil adsorbents at the adsorbent. Prashanta and co-workers, they found that in Bankura district, the fluorosis is a major problem worldwide. And Hafsajan and co-workers, the aluminum oxide nanoparticles were used as adsorbents. Zhang and co-workers, the low-cost, high-capacity iron oxide adsorbent was treated with ethanol. Al Hassan and co-workers, they used alumina and alumina-based composites for fluoride removal. Bhomik used fish scale dust and powder as a good adsorbent. Patriciano and co-workers, they used the electrodialysis reversible method. Gan and co-workers, they used the coagulation method for the removal of fluoride using zirconium tetrachloride. Ma and co-workers, they used two lanthanides-based MOF, that is cerium and europium. And Rabupalli and co-workers, they used low-cost active carbon derived from barks of Ficus racemosa, that is fig tree. So concluding my presentation, I would like to tell that the fluoride has both beneficial and detrimental effects, but yes, we should keep in mind how much fluoride we are intaking in our body, and also the most cost-effective method should be used for the defluoridation. Thank you. Thank you for your patient hearing.
Sukriti ma'am, I have one question. I mean, you have mentioned many techniques for the fluoride removal, right? So
yes, sir. Yes, ma'am.
Yeah, so I would just want to know, okay, according to you, which is the best technique and why?
Ma'am, according to me, adsorption is the best technique because the different adsorbents that is brick, char, it are easily accessible, and also it's cost effective. And second most important thing, that even at low concentration, the retention of the fluoride ion is very good. And also the most important thing is that the adsorbents that are used over a wide range is very cost effective. And also the method is very easy because we only need the ion selective electrode for the detection of the fluoride.
Okay.
Okay. Thank you, Dr. Supriti.
Thank you, sir.
May I request Dr. Mohammad Sajid Ansari for the next. You may stop sharing the screen, sir.
Okay.
Yeah, Dr. Mohammad Sajid Ansari, are you there?
Yes, sir.
Yeah. You have 5 minutes to present. Kindly be stick to your timeline. Okay.
Okay, sir. Okay, sir.
Yeah. You can share your screen, sir.
Okay, sir. Thank you, sir. Yeah, yes, sir. Sir, visible my screen?
Just a minute, sir. Yeah. So now you can share a screen. So meanwhile,
Sir, my screen visible, sir?
So no, till now it's not visible.
Sir, I am visible, but my screen. Yes, sir. Visible my screen, sir?
Yeah. It's visible now. So meanwhile, Dr. Usha Kumari, please be ready with your presentation. We will take you up next. Yeah, Dr. Mohammad Ansari, please.
Yes, sir. Yes, sir.
Yeah, please go ahead with your presentation.
Sir, welcome, sir. International Conference, Waste Management and Sustainable Development. I am Sajid Ansari from I'm a research scholar, Department of Botany, Aligarh Muslim University. So my oral presentation topic: effect of fly ash amendment, soil and physiological, biochemical attributes and antioxidant response of medicinally potent plant Bethania somnifera dunal, variety of dunal. Recycling industrial coal waste in sustainable manner. My sub theme: industrial waste and management and resource recycling. The introduction, medicinal plant Bethania somnifera and use amendment fly ash. Bethania somnifera Dunal is known as the Hindi as the ashwagandha, belong to family Solanaceae, and various alkaloids, bethaferin and bethanoids. Among these used Ayurvedic herb and treatment anxiety and stress. Why fly ash? I have used this experiment fly ash because fly ash heterogeneous mixture of macro and micro elements. Fly ash amendment increase both growth and yield of various agriculture crop. Use my fly ash in.
Sorry to interrupt, sir. Your slides are not moving.
Yes, sir.
Your slides are not moving. Can you please check with that?
Yes, sir. Then sir.
No, you can do one thing, sir. Please stop sharing your screen, and once again you just start share. Or else you can put it on slideshow mode.
So then.
Yeah. Now it's visible. Now slides are moving.
Okay. Yes, sir. I am continuing, sir.
Yeah. Please continue.
Why I'm using why I'm using experiment fly ash? Fly ash, the heterogeneous mixture of macro and micro elements, is potential soil conditioner and soil amendment improve the physiological and biological property of soil. Fly ash amendment increase both growth and yield various agriculture. Fly ash use agriculture better option to replace the chemical fertilizer and eco-friendly approach, which also help to dispose of thermal power plant waste. Objectives. My objective reflect effects of different level of fly ash amendment soil on growth, yield, biochemical attribute, Bethania somnifera under greenhouse condition. Methodology. A pot experiment conducts complete randomized block design CRVD under natural condition with average temperature 28 at Department of Botany. Bethania somnifera seed procured CMAP Lucknow. And fly ash study was collected from the fly ash pond of Hardwagan Thermal Power Plant, Kazimpur, Aligarh, UP, district Aligarh, UP. After the collection of soil and agriculture field, it was articulate 15 IV pressure for 20 minutes and the mix with coal fly ash volume by weight. Experimental design and treatment. Experiment effect of different fly ash level amendment to soil on the growth, yield, and biochemical attribute of Bethania somnifera. My experiment result. These showing energy EDX and SAM correction of pictures. Effect of different fly ash level amendment to soil on root, shoot, phrase weight, Bethania somnifera. And effect of different fly ash level amendment soil on root, shoot, dry weight of Bethania somnifera. Effect of different fly ash available amendment soil on root, shoot, length, Bethania somnifera. Effect of different fly ash level amendment soil on leaves per plant, leaf area and stomatal area, Bethania somnifera. Effect of different fly ash level amendment to soil or the chlorophyll and chlorophyll A, chlorophyll B carotenoids of Bethania somnifera. Effect of different fly ash amendment soil on NRA, carbohydrate and protein, Bethania somnifera. Effect of different fly ash level, cold fly ash level amendment to soil on antioxidant enzyme activity, Bethania somnifera, SOD activity and CAT activity. This showing scanning electron microscope SAM showing the effect of different fly ash level amendment to soil on the stomatal aperture, Bethania at control A and control A and amendment soil and fly ash 15% showing the A and B image. My experimental photo design, the effect of different fly ash level amendment soil and growth in 15%. Best result is 15% to 5 to 25% concentration. The conclusion, variable effect of fly ash were observed on plant growth parameter on Bethania somnifera among the 5 different fly ash level, 5%, 10%, 15%, 20%, and 25% amendment the soil. But 15% fly ash was found most optimum significant enhance the growth yield biochemical attribute Bethania somnifera. The screening electron microscope revealed the fly ash amendment 15% screening significance increasing the stomatal area and aperture. The amendment of fly ash, different fly ash level demonstrates significant increase the activity of antioxidant enzyme such as SOD and catalase in the Bethania somnifera as compared to the control. Higher level of fly ash 20 and more above were significant reduce the growth yield and chlorophyll content of Bethania somnifera. Thank you, sir. Hello, sir.
Work is very good. I have one question regarding like in your research you have taken Bhubaneswar somnifera, right? So what would be the effect on other medicinal plants? Would you like to comment something?
Hello, ma'am. Hello, ma'am. Hello.
Yeah, Sajid, can you?
Yes, ma'am. Yes, ma'am. Yes,
Okay,
ma'am.
okay. So you have done your research on Bhubaneswar somnifera, right?
Yes, ma'am. Yes, ma'am.
So I just want to know, what would be the effect on some other medicinal plant? Would you like to comment?
Ma'am, I am my research topic Bethania somnifera, but I Jatropha and other so on effect very good effect fly ash.
Hello, are you there, sir?
Yes, sir. Yes, sir.
Okay. Okay, thank you, Dr. Mohammed Sajid Ansari, for your presentation.
Yes, sir. Yes, sir.
So you may stop sharing your screen.
Yes, sir.
So may I request Dr. Usha Kumari for the next round of presentation?
Okay. Thank you, sir.
Yeah, thank you, sir. So Dr. Usha Kumari, are you there? Dr. Usha Kumari?
Hello.
Yeah.
Hello, sir. Myself, Dr. Rajkishore, one of the co-author for this presentation.
Sorry.
Hello.
Yeah. Tell me, sir.
Sir, myself, Dr. Rajkishore Sharma.
Okay.
Sir, I will be presenting presently. Ma'am has gone to the laboratory.
Okay.
She is taking care of COVID laboratory, and she has been just asked to go there. So I will be presenting on her behalf.
Okay. So please share your screen. Just a minute.
Just 1 minute, sir. 1 minute.
Yeah. Meanwhile, Dr. Kishore Dhanwante, please be ready with your presentation. We will just take you up next. Okay.
Okay, sir.
Okay, sir. Ms. Usakumari, are you there?
Yes, sir. I am not able to share. Just
sorry.
I'm not. I'm just trying to share it, sir.
Yeah, please. So are you facing? Yeah, are you facing any problem?
No, sir, I have just yes, initially I was facing problem. Yes, sir. Now I am sharing my PPT.
Okay.
Is it visible, sir?
Yeah. Is it visible? It is visible. So you can start.
Good morning, all of you. Myself, Dr. Rajkishore Sharma, working as senior resident in Patna Medical College, Patna, in the Department of Microbiology, and I am presenting this on behalf of Dr. Usha Kumari, scientist working in the Department of Biochemistry, IJMS. My topic for today's presentation is biomedical waste amid COVID-19 pandemic. It is a serious threat to the public health and the environment. We all know for the last 1.5 years, we are all medical fraternity and everyone is struggling with this COVID pandemic. Apart from social and other problems, in the field of microbiology or for the medical science, enormous generation of this biomedical waste has been a big challenge to the medical fraternity. Biomedical waste refers to the waste generated during the diagnosis, treatment, or immunization of human beings or animals, or in research activities pertaining or in the production or testing of biological or health camps. There has been a continuous increase in the biomedical waste generation in India from 476.18 metric ton per day in 2014 to 587.99 metric ton per day in 2020, as per Central Pollution Control Board, New Delhi. The spread of COVID-19 in India led to sudden outburst of production of biomedical waste, which included waste generated from hospitals, quarantine, or isolation centers and testing laboratories. The generation of waste rose from 3,025 metric ton per month in June to 5,597 metric ton per month in October 22, when we were facing the peak of first pandemic. It is not. The COVID-19 waste include PPE kits, masks, gloves, cotton swabs, shoe covers, needles, syringes, which were highly infectious. Traditionally, we used to believe that only 15 to 20% of biomedical waste were considered infectious in nature. However, during this COVID pandemic, everything that was being generated from isolation center, hospitals, everything was having potential of transmitting the infection. As we all know that touch is one of the source of contaminating or acquiring infection from this COVID-19 virus. So this pandemic gave us a chance to rethink whether it is only 15 to 20% of the biomedical waste that are infectious, or at times it could be 80 to 20% also. This is the image showing different kind of waste being generated in hospitals. Again, this is another picture showing injectables, tubings, plastic materials. The bulk of the waste generated during this testing of COVID testing or during hospital care, while most of them were the plastic material, which is very serious to tackle. Although all the PPE kits and other biomedical waste were being segregated, but there are chances of mixing which is very high. We segregate at the source according to the biomedical waste disposal 2016, where all the recyclable materials were being put in this red bucket. Then body parts like placenta, pathological waste, body parts were being put in this yellow bags. This is the another slide which is showing specifically for the waste generated during COVID-19. So solid waste like PPE kits and masks are being put up in this red bucket. Syringes, gloves, and plastic webs in this blue bucket, and needles and other glasswares in this transparent white bucket. Mixing of biomedical waste with general waste, especially at home quarantine facility, isolation, quarantine centers, has compounded the problem. The situation is likely to get worsened with the provision of home-based self-testing of COVID-19. As we all are aware of the fact that the government has given permission for home-based testing of COVID-19 using antigen kits available in the market over the counter. People, when we are not able to segregate our dry and wet waste in a different bucket, it is very unlikely that the kit which will be used for COVID-19 testing will be put or segregated accordingly, or it will be managed as per the provisions laid down. There is huge lack of training and awareness among health workers also, which result in faulty segregation of biomedical waste at the source of its generation. Healthcare workers involved in the management of these waste are also at a very high risk. Mixing of biomedical waste with general municipality waste imposes further risk to health workers, municipality workers, waste pickers, and waste workers, and overall to the whole community in general. The risk is not limited to the spread of only COVID-19, but may lead to needle stick injuries causing hepatitis B, C, or HIV/AIDS. There are several challenges in the management of these biomedical waste, which include lack of adequate infrastructure.
Sorry to interrupt you, sir, but can you finish in 1 minute?
1 minute, yes, just 1 or 2 slides more, ma'am. Also, improper and inefficient treatment of these waste will lead to release of toxic materials in environment, causing air, water, and soil pollution. Management of excessive amount of plastic materials produced as part of COVID-19 waste is a serious hazard to the environment. There is an urgent need to increase awareness among health workers to reduce biomedical waste production and proper segregation of these waste. Also, active community participation is required to mitigate the problems associated with it. There is need of regularly revising biomedical waste management policy and the standard operating procedure. We need to upgrade our biomedical waste treatment facilities and equip them with latest available technology for the environment-friendly processing. This is the last slide, ma'am. Challenges before us are the lack of adequate infrastructure, improper and inefficient treatment of these waste lead to release of toxic materials, and management of excessive amount of plastic material. These are serious challenges before us. We will have to increase awareness among health workers also, active community participation, and improvement of our biomedical treatment center. Thank you. Thank you, ma'am.
Thank you so much for your presentation. Dear audience, do you have any questions? Okay, so yeah, thank you, sir, for your presentation. Now we would move to our another presentee. I'll request Dr. Kishore Dhanwate. Please share your presentation, and I'll request sir from Usha Kumari to just stop sharing.
Yes, hello, sir. Hello. Hello, sir. Meeting.
Yes, ma'am, I have stopped sharing myself.
Yeah, thank you so much, sir.
Hello. Hello. Hello.
Yes, sir.
Can you hear ma'am?
Yes, sir, we can hear you. You can share your slides now.
Can you see? Can you see the PPT? Hello, can you see my PPT, ma'am?
Not yet, sir.
Yes. Wait. Okay. Back. Okay, we will send it. Will take Zoom. Yeah. Document. Wait, ma'am. Document.
We cannot see your screen.
Hello. Wait, I'm trying. Just a minute.
Okay, no issue, sir. Sir, can I help you? You just go to your slides, just minimize it, and then come to Zoom link. You can share your screen by just going to that PPT.
Right now. Can you see now?
No, sir, not yet.
Just a minute. Yes, sir. Link one. Yes, sir. Meeting is there. Back. Share. Share to click करो सर. What should I do? Screen. Just a minute. Start now. Start now. Click करो. पुढे केलं. Start. Back. बरोबर. याच्यात कुठे आहे आता? Back. सर एक मिनिट. एकच मिनिट. स्क्रीन क्लिक करायचं होतं. स्टार्ट नाव क्लिक करायचं. आता क्लिक केलं ना. मटेरियल पुढे येईल. एक मिनिट. हे जरी केलं आता पुढे येईल. मटेरियल करा.
I think you are having network issue too. So I think we are not able to see your screen.
Okay, just now, right now?
No, sir.
Not looking.
You
madam,
you might have.
I'm trying again.
Okay, no issues. So we'll take another presentee. Dr. Karishma Shah, are you there? Dr. Karishma Shah, can you hear us? Dr. Mayank Singh, can you hear me?
Yes, ma'am.
Okay, sir, so can we proceed with your presentation first?
Yes, ma'am.
Okay, sir. So I'll just make you the co-host, then you will be able to share the screen. Yes, sir. Now you can share the screen. Dr. Kishore, we will take you after Mayank Singh, sir.
I don't know why it is showing unsupported.
Sorry, sir.
It is showing unsupported.
Unsupported. Let me check. Sir, you are the co-host. I think you should be able to share the screen.
Okay, ma'am. Just wait for a moment. I am connecting.
Just try by your end.
Okay, ma'am. Bye. अरे
Sir, you are facing error while opening PPT or while sharing sharing it. Yes, sir, we can see your screen, but I'll request you to keep it on slideshow mode. Yeah. Yes, sir. You can proceed.
Hello. Hello.
Yes, sir. Yes, sir. You are audible.
Can I start?
Yes, sir. We can start.
Okay. Good morning, everyone. Myself, Mank, working as a project assistant under supervision of Dr. Pooja Khare, CSIR CMAP Lucknow. Today, I am here to present my oral presentation, and my topic is application of biomass-based derived biochar for removal of anionic dye from water. As we know, water is a vital source for ecosystem as well as for human being. But due to some pollutants, its toxic effect, the flora and fauna in the environment are disturbing. Among these contaminant, dye is one of the common pollutants from textile industry, which deteriorate the physicochemical properties of the water. And the molecular structure of dye is very complex, and it persists for longer time. And for removal of the dye, the adsorption is one of the best technique, and biochar is a good candidate for absorption. So we can see the effect of dye pollutant on plant species, as it interrupt the photosynthesis activity, decrease the soil quality, affect the plant growth. On aquatic, it increase the COD, VOD, and other components. And in human being, it causes carcinogenic effect and causes cancers too. The main objective of my study are characterization of biochar prepared from different biomass, and to evaluate the absorption capacity of the biochar. And third one is the phytotoxicity of gram seed. Here we have taken 4 type of biochar: sugarcane bagasse biochar, which is represented SBB, coconut biochar, which is represented CNB, and PDB as paddy biochar, lemongrass as LGB biochar, and Ramadan brilliant blue dye. And on the basis of seeking time, temperature, and pH, the adsorption of these 4 biochar is done. You can see the results, the different concentration of dye 500, 600, 700, and 800, 900 ppm were prepared. And after adding biochar, these 3 things, these 3 parameters, pH, and on basis of seeking time and on basis of temperature difference, the removal of dye effluent is done. And you can see the result under this before and after treatment. And some physicochemical properties were also done, like pH, EC, carbon, total hydrogen, and nitrogen. Here you can see the images of SEM and FTIR, as we have done the SEM. And in SEM, the biochar irregular structure and micropore are in tubular arrangement. And in FTIR, mainly 5 peaks are observed at 3427, 2924, and 2834. Here we can see the agitation time graph on the basis of 5 minutes, 7 minutes, 15 minutes, and 20 minutes, in which the coconut biochar is, and it had absorbed 99% of the dye fluent. Here we have done the phytotoxicity test, in which the dye contaminated water, and after adsorption, the filtrate is taken in which the gram seed were grown. Germination index was very good in both, but we have seen the stresses in the dye treated and after filtration. After filtration, there were no stress, very little amount of stress as we generally seen when we grow in soil. And the recycling of biochar is done up to four cycle. The recycling was done. First two are very good. After that, it reduces. Now I'm coming to conclusion. Biochar prepared from different biomass uh, have good capability of removal of dye, and the coconut has absorbed 99.47 to 99.99%, then sugarcane bagasse biochar, then lemongrass biochar, and then paddy biochar. The recycled biochar uh, 4 cycles, and no phytotoxicity was observed, and the sludge, the dye sludge after filtration can be used as a soil fertility enhancer. It is very good. We can use for soil conditioner also. Thank you.
Thank you, Mayank Singh. I have one question. I mean, what
Okay,
is utility approach of your research work?
Yeah, ma'am. First, we can purify the water before discharging into the water body from textile industry. First, at very low cost, and second one is after filtration, the dye sludge which we obtain can be used as a fertilizer also. It will enhance the growth parameters in plant, and make the soil best for germination also.
Okay, okay. So what are the advantages of biochar over this water purification technologies that we are having right now? So
First, it is low cost. Then second is it prevents from burning and wastage of crop residue. We can utilize the waste in very useful manner, that it can be used as a adsorbent as well as for soil conditioner.
okay, thank you.
Okay.
Excuse me, Mr. Mayank.
Okay.
Am I audible now?
Yes, sir.
Okay, so what is the yield you have got?
What is the
yield of biochar?
Yield of biochar. If I taken 5 kg of raw biomass, means after harvesting the crop residue, if I obtain 5 kg, then it's 40 to it depend upon, firstly it depend upon the biomass. If biomass is highly producible, then 50%, either 35 to 40% of the biomass is converted in biochar. If we taken 5 kg, then biochar will be near about 2 to 2.5 kg.
Okay, thank you, sir. Any questions from the participants, audience?
तिकडून काय होते शेअर केलं का थांब की अजून आलो नाही.
Okay, Mayank sir, you can leave. You can stop sharing the screen. I'll request Dr. Kishore.
Okay, thank you, ma'am.
Welcome. Dr. Kishore, I think there's poor network connection from your end.
Hello, ma'am.
Hello, sir.
You start your screen sharing now.
Okay, just a minute. Let me make you the co-host. Yes, sir. Now you can share the screen.
1 minute. Huh?
Are you able to see screen, ma'am? Hello? Hello, ma'am.
Yes, sir.
Are you able to see, ma'am?
No, sir. No, sir. I am not able to see your screen. Okay, one thing we can do, you can email us and we will share you on your behalf. I'll just, yeah, you just try by your end.
Okay. Okay. Email send करो sir. Check it. कौन सा mail है sir? Mail कौन सा है? Mail विचारा. Madam, give the mail please. Hello, ma'am.
Yes, sir.
Give your mail please.
Yes, I'll drop in the chat box. I'll drop in the chat box. Okay, so we'll proceed towards our next presentee. I'll invite Ayushi Varashne to present.
Yes, ma'am. Can I share my slides? Hello, ma'am.
Yes, you can share your slides. You are now co-host.
Okay, ma'am. Sure. Are my slides visible, ma'am?
Yes, it is visible. But again, I'll request you to
slideshow.
Slideshow, yeah. Yes. Now you can proceed.
Okay. Good afternoon to all of you present here. My name is Ayushi and I'm doing my PhD from Amity Institute of Biotechnology, Amity University, Noida, under the guidance of Dr. Sumedha Mohan and Dr. Praveen Daya. And I am here to present some of my work related to my PhD on the topic eco-friendly utilization of fly ash in agriculture for sustainable development. Now, the question arises, what is fly ash and why fly ash is a major concern all around the globe? First, let me start with the introduction that what is fly ash. Fly ash is a fine particle residue produced from thermal power plant every year. It is a major pollutant that is generated from coal combustion process for the generation of electricity, and it contains huge amount of alumina, silica, and small unburned carbon. Fly ash is an amorphous mixture of ferroaluminosilicate minerals. Fly ash has certain properties, including it is lightweight in color. It has lightweight gray in color. Particles are composed of fine glass and spherical in shape with a size range of 0.5 to 100 micrometer. Now, why it is the major concern all around the globe due to the presence of toxic metals, which causes environmental pollution and increases the risk on all life forms. But due to the presence of unique physicochemical properties, because fly ash. Yes, ma'am. Hello. Hello. Ma'am, can I continue? Hello. Am I audible to all? Hello. Is anybody there? Hello.
Madam, you can proceed. Yes, you can continue.
Okay. Now, due to the physicochemical properties of fly ash can be utilized in agriculture with certain limits. Now, on the next slide, on the ma'am, someone has shared something here.
Yes. Just a minute. Yes, just a minute. Ma'am, can you? Yes. Madam. Ma'am, sorry to interrupt, but can you? Yes. Just a minute.
Excuse me, ma'am. I would request she's presenting and who else?
Having some technical issues. Now sorted. Ayushi ma'am, your internet connection is unstable. I'll request all the participants to please join by the meeting link provided in the chat box. We are facing some technical issue with this link. Dear participants, there's a meeting ID. It's delayed. It's very delayed. See, मीटिंग लिंक इसमें नहीं है,
इसको लॉग आउट करके लॉगिन कर सकते हैं।
कर सकते हैं।